Record. Oh, sorry. Anyways, um, so we're just going to go over each angle, like boop, 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 and see which one is the most energy efficient. When it comes to modeling and when it comes to anything in science about bonds forming, the easiest way to think about it is how is it energy favorable, or what's the lowest energy point, because that's how things are going to form. Maybe we can look at graphs on that. Maybe they're in the slide, but that's fine. The one we're doing is template-based modeling. We already have a template. You guys know what your template is. Your template's what's in your the patents. So you can just do that template-based modeling and just put your sequence on it. And I'll show you how to do that also. But I think it's good not only to just do it, but know what you're doing. Because it's also, you know, I know y'all aren't just doing this to plug some things into a computer, but y'all want to learn, hopefully. It's okay if you don't. I understand that too. There's some things I did in high school where I just did them to do them, and I didn't really want to learn because I already learned enough. So why? You guys know why. I'm not going to really go into that. How are they done experimentally? Very big machines. Okay, so I guess I have a question for you. Why is physics-based modeling? Why is it hard? If I don't know, yeah. Why can't you just do physics-based modeling for everything? Because that would, you know, make sense. Why can't you? I guess. I'll pull up the chat. Any takers on that question? No. Yeah, lots of yeah. That's exactly right. Thank you, Emma. Yeah, much complicated. Very wow. It's going to be hard. It's going to take hours upon hours. I was actually modeling a really large protein used in E. coli bioenergetic stuff. They're part of one of those pathways. I think it took like 2 weeks because I needed to do a physics-based model because I don't know, I just needed to do it that way. It took 2 weeks to run it on like a collection of computer clusters. You can't run these on your own computer. If you try to run these on your local computer, your computer will die, or it's going to take longer than time we have in the universe. So it's not going to work. There's too much going on. So you can't do it there. Pretty much what they're saying. Timescale won't work. So knowledge-based methods, and that's how we're going to do it. They're data-driven. We talked about that. You know, we know these experimental models, pretty easy. We have this data, so why not use the data to form these like heuristics? You know, we can base the modeling off this data. So similar sequences tend to have similar structures, hence why we're doing homology modeling. Some cool graphs here. Don't worry about them. All right, you guys know PDB, or you should be at least somewhat familiar with it after this project, which is actually really good because, I mean, this is the upper level class in college that I'm taking as like a junior, and they're now just introducing to you what PDB is. So that should make you feel very smart because you are, and that's a skill set that a lot of people don't have. So, okay, let's talk about template-based structure prediction and basic workflow. All right, so you're going to hear these things. You're going to have a query sequence with an unknown structure, so the one you have a question about, and then we want a template. I don't know, when I was first learning this stuff, it kind of confused me, like query template, what the hell does it all mean? They're also called, query is also a lot of times you'll see called target template. I think that one's a little less clear, but yeah, regardless, query is the one you have that you don't know the structure to, and the template is what you're basing it off of. I'm just kind of stupid, so it took me a while to really, like, I'd get them messed up all the time. So you're pretty much just building a model on the template. And, you know, think of it like anytime you bake and you have a mold, you're pretty much just pouring the sequence into that mold, maybe making cute taiyaki or something, I don't know. So what does it mean for two to be sequenced? You guys know that. You guys can answer that question. We have sequence identity. Whoa! So yeah, it's just sequence identity. That's kind of really what it means to be Coolio. Don't worry what their tools they're using. I kind of want to show you some like models for this. We're going to be using a different tool. I don't know how similar. They actually are pretty similar. Yeah, don't worry about it. I'll show you how it works anyways later. But ab initio modeling. Go to our YouTube page, and I did a webinar on ab initio structure prediction and stuff like that, and how to set it up with Rosetta. So you guys can go look at that. It's really boring if you're not into this stuff. But hey, if you are into this stuff, you can check it out. We won't be doing it really at all. So don't worry too much about it. De novo structure prediction. Latin means like, I don't know, of new. I don't know. But I don't speak Latin. Fragment assembly. Okay, so this is what we're going to talk about too, is fragment assembly, and I'm going to have you guys get fragments. So fragment assembly as a method, I'm trying to think of the best way to explain it. So you get these like MERs. So you hear 3 MER, 9 MER, whatever MER. And this is we have data. We have empirical data on certain sequences within structures. So, okay, sorry. Where's the webinar on the YouTube channel you mentioned? I'll show you. I can actually show you in a second. Let me pull that up. And so just make sure you go to YouTube. Alrighty. So cool. Sorry. No, that's not what I wanted. Command C. Oh, there we go. Colin knows how to type. Go here. I think that leads to the right place. Yeah, it does. Dope. And subscribe. But yeah, if you go there to what the link I posted, and let me post the exact video. Sorry, I'm kind of scatterbrained right now. If you can't tell, I don't have a script for this or anything like that. I really was just going to do what I do, which is not always the most efficient. So, you know, here's the video. It's called Protein Folding Rosetta Commons Tutorial. I have short hair in it, and that's always awesome, having short hair. And that's the video right there. Thank you for being interested. Yeah, no problem. Let me share my screen again. How do I do that? And you see all my NBA things because the finals are going on and the Lakers are going to win, which is big sad. Sorry if you're an LA fan, but I am not. And the Lakers are awful. Just kidding. Ha 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 ha. All right. Where was I? De novo structure. Wait, let me pull out my chat again. Cool. The chat is up. It's just like you pretty much like de novo synthesis. You're just kind of pulling things at random. Rosetta will do that. And that one is the one I was talking to you about earlier, where you just kind of you'll just incrementally do each angle turn in the bonds and see which one is the lowest energy. And then that's your protein prediction. That's useful if you don't have any idea of what this may look like. So if you are doing a completely novel structure, no one knows what this protein looks like, you just have the sequence, then it's good to use. If you're not doing that, don't do this way. And so you can also use fragments where, let's say we have these like pack of 3 MERs or 9 MERs, so these 3 residues or 9 residues right next to each other, or amino acids right next to each other. We have data on how most likely these will form. This is a really good way to predict your secondary structure, so your alpha helices and your beta strands or beta sheets. But it's really bad at predicting. It's only good at really predicting alpha helices, not really good at predicting B strands. And the reason for that being is because alpha helices, they have about a 4 residue offset. And so if you have a 3 MER or 9 MER fragment, you're going to be good at knowing that 4 residue offset because you have that data. But B strands can like kind of form across proteins and whatever. And so they're going to be really bad at predicting that. So this is better than just doing de novo. It's especially quicker because your algorithm can base it off that, and it's going to give you more of an idea of what the secondary structure is, but it's going to be really bad at predicting certain aspects of your secondary structure. Also, just type in chat like Y or N, I don't really care, but just give me a signal if you have or have not messed around with PyMol before. Because that's going to be important and inform me on whether or not I need to show you how to do that. Yeah, dope. Everyone type in yes. Yeah, woo, yeah. All right, cool. that saves all of us a lot of time because that was going to be a mess. So yeah, cool. So if you guys have PyMol, one way to do it, do you guys know how to get your RMSD scores in PyMol? So RMS score is like root mean square deviation or something like that. It's just pretty much the difference between your protein and your and another protein. If you haven't done that before, I can maybe show you guys later, or I'll just send you guys a picture of how to do it. But yeah, I'll show you guys that later when we do the whole shabango. All right, so we'll use Rosetta. We will also, or I'm kind of debating on using Rosetta right now or showing you guys Rosetta because it's a little more complicated. But instead we may just do Swiss model, but we can also use Rosetta for ab initio structure and then compare those structures. So yeah, I'll probably show you guys both. It's just Rosetta's gonna take you a really long time, probably. It's gonna take a couple weeks to get results back. It only takes like, at most, it's gonna take you like an hour to get your initial stuff, like all set up and whatever into the system. But then to actually get a result, which doesn't cost you anything, you just gotta like update and see every once in a while. It may take a while, depending on the size and the complexity of the protein. They do knowledge-based strategy, yeah, because they use the fragments, right? They also have their energy function. Don't worry about that. We're not going to do math because we don't need to do math. It's kind of based off of the Boltzmann equation, if you guys want to do research on that, but they have other things that they do. And yeah, don't worry about that. It works on potential energy. Like I said, pretty much a good way to understand energy when it comes to proteins, at least right now, is just the lowest energy is the best energy. And if you can get your protein to a really low energy, that means it's a good protein. But as I'll show you, there's a way to do that. There's a really cool thing called the Monte Carlo sampling, which just kind of goes residue by residue and will search for the lowest energy. Metropolis criterion is a Monte Carlo method. Don't worry about this. It's just, I don't know, look up that stuff if you're interested in coding. It's a really easy coding exercise if you know how to code. And you can implement it yourself. It's gonna suck. It's gonna suck real bad, but it's cool to practice and do, and helps you get an understanding of what maybe how some of these computational things work in a very simplistic way. I think that's kind of it. I'm just gonna make sure. Oh yeah. What's up? All right, yeah, that's pretty much it for that stuff. So that's just to give you guys, so sorry, Mahi, I don't know if you're asking a question, I kind of cut off.
Oh no, I'm good. Sorry about that. I was just back.
No, you're good, you're good. So yeah, that's just the background information, always necessary to understand. So if someone asks you, what are you doing? You can say what you're doing. And it just makes more sense. Alrighty, all right, give me a second, I'm gonna pull up a couple websites. And we'll get started. If you guys have any questions or whatever you want to share, maybe something about yourself, please go ahead, because I don't know most of you. And I'm Colin. And I'm from the water tribe. We're a Swiss model. All right. Sorry, a second. You're gonna get an awkward silence. One more. Yeah. Dope. Okay, cool. Now I'll send you guys. Okay, cool. Oh, I'm not screen sharing anymore. Yeah. And here we are. Let me also pull up PDB. Protein data bank. Cool. So here we are. Yay, look at us. So there's a couple websites here. We got Swiss model. You can just search up Swiss model or go to this link. We got robetta.org and we got robetta.org, but they're going to do two different things, which is pretty dope. So let's start with Swiss model. Swiss model is really, 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 really easy to use. And it's really quick. And in fact, I bet it'll be done generating by the time I explain the other two websites I linked there. So we got a target sequence. So what's our target sequence? I don't know. I don't know. I don't have a target sequence. Let me go to PDB and like find a target sequence. So I can go to beta. Yeah. Let's do this one. This is just the one off the top of my head. You can come here. You guys have your sequences generated, so you don't need to do what I'm doing right now. Let me get the sequence. Sorry. Please. There it is. That's my query sequence. You see, they even named it for me. So Command C, we go back over to Swiss model. They got this little neat thing. You paste it. Now, oh, and they'll do whatever that color thing is. I don't know what that is, but don't worry about it. So you can add tags and stuff. Don't worry about that. They have a really nice thing. If you don't, you guys use, I would recommend using your model, but you can also search for templates too. And this is going to search all the protein data banks for the templates to use for this. And so it's going to find the one that matches it most closely. And then usually what you want to do is model it after the one that has like the closest match to it, because that would make sense, right? So I don't know. I recommend trying. What you can try is either query. What are the templates for? These templates for? Great question. Give me. So the templates are what the protein's going to be modeled after. So you know, it's like the mold. So your protein sequence that I just copy and pasted, the one that they're looking for templates for, is like the batter, and you're pouring it into the mold. And so this is pouring into the mold. It's looking for the templates. And so you have a query sequence or a target sequence. Those are synonymous, which is what I just pulled off of PDB. And this is the thing. Let's say I don't know what this model looks like. That's a good way to think about it. The query or target sequence is you don't know what the model is. This is what you're asking questions about. The template sequence is we know this model, so we can use the query, or we can use it for the query target sequence to model after the template sequence. Does that make sense? Dope. So yeah, so this can search for templates in the best template for you. Most likely what it's going to pull up is exactly this. And so it's going to make a real nice model for us, because that would make sense. But it may pull up other things, and we'll see. And I'll show you what it does when you do that. This is going to take a couple minutes. The Swiss model, like I think at most is going to take like 30 minutes, 40 minutes to run. And that's like at most. So especially if you look for if you don't look for templates, you have your own templates, it's going to take less. But like I said, I recommend when you guys are doing your protein sequences that you generated on your own, search for templates, but also use the templates that you know, that you evolved from. Does it make sense what I'm saying there? Like use Swiss model for both. Cool. Yeah, if you guys don't know what I'm fucking saying, like just let me know, and I'll clarify, because that's kind of, I don't know how to word that better. So we got Robetta here. So this is also going to do a more complex structure prediction. So Swiss model is a little quicker in the way they do it. This is more complex. Don't worry about the coding. Maybe we can have a separate, if you have any questions about that, just, you know, message or something me, and we can talk more about that, but it's really not important right now. It's more important on how to use these tools. But you remember what I said about the fragments. If I just enter in a protein in a template, it's not going to really work. But if I do fragments, that's going to be really good. And so what you're going to do, you need to register something like that. Just literally all you need to do is name or email. I just do my name. You get a target name. This is the same one I do. you can paste the FAFSA. FAFSA is just a bioinformatics file format. So think of it like a TXT file, but for bioinformatics. And there's a little more information baked into the file. So bioinformatics software knows what's cooking, what the hell is going on with this. So you just paste it like I just did here, and it's going to know what to do with that. And then this website here, old.robetta.org or robetta.bakerlab.org/fragment submit is going to generate those fragment files I talked about, which are these shortcuts on how to fold the protein in a way is thinking about. These are we know what these 9 residues, when they're next to each other, we know what those are. And then you submit. And yeah, and then your job, you'll get this little thing, your job is added to your request queue. Click on this job idea and you'll know status queued, whatever. This is going to probably take about a day or two. And so sorry about not having that generated. Maybe I should have done that beforehand. This is a decently long protein, so it's going to take some time. And this is going to generate a ton of data for you that's going to be really necessary. But yeah, you're going to get these files. I'm trying to remember the name. Maybe I can show you the name of them. Give me a second. And maybe I can kind of give you guys a better idea of what these files are going to be called. Because that would be good, I think, I should have done that. But hey, we live and let die. Boop, 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 boop. Okay, cool. I don't know what the best way to show you this is, but yeah. Wait, let me see if I have something queued. Because I've done stuff before, but it's probably like not. Whatever. It will be popped up. And like the files, pretty much what they look like is, let me share this. This is gonna be a bad way of showing you guys, so I apologize for that. But so here are my files. Great game if you haven't played it. These are what the files are gonna look like. This is the one ELWA protein. I don't know what that protein does. I forgot. But also watch the video that I made on YouTube, and I talk about these files a little bit more. These are the fragment files. So you're gonna want this one. It's like a V13, V13. If you see those are your 3-mer fragment files. Again, the fragment files are just the short fragments, and we know kind of what they do. And then you want this SS2 file. Sorry, I see a chat notification, so I need to pull that up. Where's the YouTube channel? I linked it above. I can't copy and paste. That's so stupid. I'll send it again in a second. So yeah, so you get these the SS2 file is more of a 3D kind of structure. So once you're past your secondary structure, you need a tertiary structure. Oh, thank you so much, Emma. And Knock. Thank you guys. So you get the V13, the V13, and you can see here, sorry, ELWA 03 and 09. You want both of those because it's 9-mer and a 3-mer fragment file. The 9-mer has a little more data, but the 3-mer is a little more specific. You can see those the importance. Just a different search queue. And then you want this SS2. So you download all those 3. Is that clear? That's what you see. You see this 09, this 03, and this SS2. And those are going to be your prediction, like help you predict the structure. If that's not clear, we can talk about it too to make it more clear. Share again my general screen. So this will generate those for you, this little thing. And you'll once your job's done, it's active. See, it's doing it now. It'll take some time. So, okay, now you generated your fragment files, and we can see we have those downloaded on my computer. Sorry, let's see. Wait, what is Robetta for again? Yeah, no problem. Robetta does 2 things. So the stuff you're gonna see on the Robetta runs Rosetta for you pretty much. Rosetta Commons, not Rosetta Stone, even though it could be the same thing. I guess they both do translation. Rosetta Commons does it does that ab initio structure protein. It does a lot more, but it does that ab initio protein folding that I was talking about earlier, which is a little more specific protein folding. It's a homology model, but it does a little more specific stuff. It's a more complex algorithm. You can use Robetta. Robetta is developed by Baker Lab, which I forget what university they're at now. Maybe up in Washington. Robetta is a software that does Rosetta for you. So you don't have to worry about downloading Rosetta and stuff like that. You can just go on Robetta and do all that, which is really nice, as you can tell why. So there's two websites. There's the Robetta, oldrobetta.org. And the way I find this one is Robetta fragment generator. You can just look that up in YouTube and you'll find this website. Yeah, I'll explain what you input and what comes out in a second. For this website, all you need to input, excuse me. Let's go here. Yeah, for this website, all you need to input is you need to register a username. So whatever, make your username, they won't care. You need a target name. So like, what should we call it? You can literally put in protein. Doesn't matter what you put there. The thing that matters is you put in the sequence here. So what I copy and pasted from here, the sequence is what you input here, and then you submit. And what this old Robetta thing, this is the fragment generator. So this is going to generate your 3-mer, 9-mer, and SS2 fragments, files. And that's going to be useful in this next step we're going to do. So that's what this Robetta website is used for. This Robetta, which is the new server, this is the old server. You can't do this stuff on the new server, so you got to go do it on the old server. The new server, this is going to do the actual structure prediction. But the reason why we need the old server to generate those fragments for us is because those fragments are going to make our prediction a lot more accurate. And it's also going to save time and computational power and all that stuff. Because those fragment files are like a bunch of data that tells you tells the protein how to fold. Does that make sense? Dope. So we're going to go to structure prediction. Pretty easy. Submit. You're going to go to submit. Hey, I already made a thing. That's really nice. Again, target name, egab. Here's my file, right? We have this. So that's the same setup, but you have these files, right? And so here, look at these optional. So we have these PDB template files too. So either it will generate its own template for you, or you can put in your own template. Remember, we know what the template proteins are, because that's where you did your kind of evolution off of. That's where you did, that's where you generated all your other ones that are outside of the patent. The templates are the ones that are patented. So you just grab those PDB files and you can choose those. And that's what you do for there. And then you're going to come here to Open Fragments panel. And this Open Fragments panel, you can see you can download the SSS, and then you can add other fragment things. So the SS file, sorry, the SSS, no, just the SS file, is that one I described earlier that helps tertiary structure. Yeah. And so I recommend just putting in like that file there. And you can add PDBs. Yeah, you don't need to do that. And then you just need to answer this to make sure you're not a robot, and then you're good. You can choose keep private or not. It really does not matter. Yeah, so your FASTA is this FASTA thing is the one you generated on your own from the PDB file that the protein is patented other, and the SS file you're gonna get from here. Now, if you watch that YouTube video I did earlier, you can run some of those own sequences on your own computer. You need the 3-mer and 9-mer fragments to do that. Feel free to do that. Don't run a 1000N, maybe run like 10N or something, and you can do that on your own. But yeah, they're going to generate those fragments also for you here. But to get the SS file, you need this, which is just going to make the tertiary structure more predictable and easier to generate, which is going to save us time. So you submit that. After you do this, right, you got to submit that 5 thing. And then it's going to be a very similar thing. You're going to have a job ID and a queue, and then you can just look up that job ID or queue whenever. Every time you come into here, you can sign in. You can see under your name, you say, oh, my queue should be empty. Yeah. So mine's empty right now because I didn't submit anything, but yeah. And you guys can be taking notes on this or whatever. I'm also recording this and I'll post it in the general chat after. How long does it usually take, and what do the results look like? That's a great, that second question there. When you guys generate results, maybe we'll have another kind of meeting like this where I'll talk about the results you generated and tell you if they're good or not. I don't have that right now. Pretty much they give you a bunch of data, and I'll show you a little bit what that looks like with the Swiss model stuff, because Swiss model runs a lot quicker. How long does it take? It really depends. These are a little more complex. They don't run on a supercomputer, but they have like crowdsourced computer clusters that people will like donate to them. They'll donate computer hours so they can run these like very complex algorithms. It can take anywhere from a couple days to a week or two. So that's why I'm saying like do this also, but while you're doing this, also run Swiss models on them so you guys can get data a little earlier and you guys can talk about that and maybe give you some clue. And then once you have these done, you guys can compare the data. The Swiss model will take like an hour. So yeah, this will take pretty long to run. And results will look very similar to what Swiss results look like. So that's Robetta. Put in chat if you have any questions about that or whatever, or how you do. So while we were talking about that, Swiss model generated the templates for us. So this two ways to do Swiss model and Robetta. We're done with Robetta. That's how you do Robetta. That's going to take a long time to run. But in the meantime, run all your templates you guys generated on your own with this. And so we have these templates. Sorry, I need to move some things around my computer that you guys can see. So we can generate these are the templates it generated for us on the model or on the sequence we put in earlier. These are probably the exact same ones because they just go through a bunch of protein data banks. So these scores here, I mean, you're just going to pretty much choose the top one. You get the 101, you get the 066. As long as this G score, I'm going to call it, is near 1, you're chill. Like if that's around 1, then that's going to be a good template for you to use. Ligands, you shouldn't worry about ligands. Again, ligands are just going to be there. And then all you have to do here is you can select the ones you want to model it after. So we're going to choose the ones that these 4 look very similar. So why not model it after all 4? And so we just click build models, and this is going to go to the screen once your templates are found, and then it's queuing. So these will run. I don't think these will take very long. You see they're already running. Swiss model is very fast. So while we're here. Yeah, so awesome. Yeah, cool. So once this runs, I guess we can then I'll show you what it looks like. So yeah. So you guys come here often? Well, it's done running. Some of them are. Whoa. Very fast. I told you it's very fast. Again, the important thing is this G score and this Q score. And they do a very nice thing. They put a little thumbs up next to it if it's chill or not. So let's look at one. Let's look at this first one. So we're going to just open up all these things. The things you really need to focus on, these scores will pretty much give you a nice overview. Once you generate this stuff, I don't know, I just like save, like, I don't know, you can take a screenshot or whatever, or just you can download this stuff, I think, and just save it. Yeah, you can download these models right here. Sorry. Okay, that's a great question. The GMQE is kind of a general score for how good the protein is structured. It takes a lot of energy, algorithms, or like functions into thought, and also how well it pairs up with the template protein. Don't worry too much about it. Just know when you look at here, if it's about 1, it's a pretty good score, and that means your protein's doing a good job. Like it's a good model, and most likely that's a good one. It's not like really present identity and blast. It has more to do with the protein actually folding. So it's the energy functions that take the protein to fold in the tertiary structure and secondary structure. In comparison to the template model. So I mean, that's really all you should worry about. Does that make sense? Yeah, cool. And so the important thing, if it's near 1 within like a couple 1/10% or 1/10 of a number, it's not a percent, then you're doing great. And they'll tell you, right? This blue is good and the white's good. So all this stuff, this is a good model. And we could go on with this one and we could talk more about it. And maybe even use, let's say you wanted to generate more templates, we could use this template in Robetta to also compare. So yeah, good model here. You don't really, this is model template alignment. It's exactly what it sounds. It's how does your model align with your template? And pretty much that's all you really need to know. The sequence identity is 100%, by the way. So this one's going to get it really good. This is like, you're not going to get these scores with everything. And you can see this here. This is a good, if you pull this up, let's read this graph. Protein size versus a normalized Q mean score. So this is pretty much, again, how well does it score? And so our protein size is rather large. It's 550 proteins. But and as you get larger, there's not many proteins like this, but usually they're not very accurate. But ours is extremely accurate and, and rather large. So there's not a lot of room for error, but we did a great job with this. The reason why is because we knew it was going to do very well. So this is almost like a sanity check that this system works. Yeah. And that's pretty much it. So 2 things, right? That's how you run this. Run them. Run your targets, your target or query selections on these templates. Maybe you generate in Swiss model or that you have already. And then while you do that, you can run these Robetta stuff in the long term. So yeah. And Robetta, again, is going to take a while, while Swiss model, like we just demonstrated, took less than this entire thing. And once those are generated, we can maybe have another little meeting and talk about which models are good, which models are bad. I can take a look at them. I can tell you why or why not they're bad, probably. And you guys can do a little digging yourself. How many models do you guys have to need? How many models do y'all have to go through, would you say, per team? Just type in chat or something. Like how many sequences do you guys generate yourselves, would you say, per protein you're interested in? 16? Okay, dope. So yeah, I mean, you can create a huge queue too with the Robetta. So I just queue those up all at the same time. And then Swiss model, you're gonna have like probably about 32 things you generate, because you wanna generate one where you search for a template and then generate one which you insert the template. So 32 for those, and then 16 here, so you have 48 in all, when all is said and done, I guess, that you can really talk about and look at. And yeah, the real time limiter is the Robetta stuff, but we can meet and talk about the Swiss model stuff too, whenever that's done, because that's going to give a good indication of what is and is not working. I imagine most of your proteins, if I don't know if the other groups told you, did y'all just kind of take proteins out randomly, or did you guys take like the hydrophilic proteins out and replace them with like other hydrophilic proteins? Because it's important to do, because if you just take proteins out like willy-nilly, you're going to run into problems. Any chance? Yeah, I meant mutants probably. Sorry. Yeah. By models, what I mean is like, and when I say evolution, I mean mutations. Sorry. I don't know why I got like a crux on that word. I use that as a crutch. Um, but yeah, when models, I mean like mutant ones y'all generated. So they have 16 ones they generated separate from their template that they're basing it off of, which is what's patented, is what I mean. You don't need to run the patent through this stuff. We know what the patent looks like because it's patented, right? We care about your guys' mutant strains. Similar color proteins, hydrophilic. Good, yeah. I mean, if the color, right, went both directions of hydrophilic, hydrophobic, maybe one thing you guys can do as a team, or y'all can do as a team, sorry for saying you guys, y'all can do as a team, is look at which ones you mutated. And as long as it's more of the hydrophobic side, hydrophilic side, excuse me, hydrophilic side is the ones you want to mutate, it's going to be good. And mutate those to other hydrophilic residues. The reason why we do this is because think of the way your protein's going to fold. It's going to go right onto itself. And what's around it is water, right? And so the hydrophilic proteins are going to kind of form that like shell, and they're really usually often easy to replace. And if you replace them, you're not going to do a lot of damage to either the binding sites or to the protein structure. Very often not. In fact, David Liu, who's a great researcher at Harvard, he developed a model on how to supercharge proteins, which is used for a lot of useful things, in which he just went residue to residue, and if it was on the backbone or whatever, it was like a hydrophilic one, he's like, yeah, we can replace it. And that model seemed to work very well. So that's what makes us, at least me, think that you could probably do this with the same thing, but do it by hand. And so yeah, think of it like that. So that's why you want to replace the hydrophilic ones and not the hydrophobic ones, because oftentimes, I mean, not always the case, hydrophobic ones are inside, pour into the structure, and also maybe in the binding sites, which can be dangerous because if you change the binding site, you know. But usually those binding sites are probably also protected. So yeah, that was very quick and I talked very fast, so I apologize about that. My cardinal sin. Get it? Ha ha ha. But yeah, I'm here to answer pretty much any questions y'all have, and fire away. Fire away. If you guys don't have any questions, I'll post this again in the chat, and you can always ask me questions in the chat when doing this, just to make sure y'all are doing it correct. Good, I'm glad. Yeah, I'll pretty much just stay here until, oh wow, my lighting is real shit. I didn't realize. I look like, I don't know. I haven't watched Harry Potter, but they're like Dementors or whatever. They like suck the soul out of people or suck the magic out of people. I don't know what they do. I know it's a sin to be a self-described nerd and not watch Harry Potter or read it. Done none of that. Lord of the Rings. Souls. Thank you. Very important to, hey, we are scientists here, right? It's very important to understand the proper function of Dementors. So thank you. Alrighty, yeah. Feel free to leave if you have questions, and maybe if you guys don't want to ask in like the public chat, just DM me here or whatever. Yeah, and that's pretty much it for Robetta and Swiss model, which is how you're going to generate your protein models initially in order to see if they're going to probably work or not, or they're worthwhile going through, and which ones to use. Yeah, of course. Bye.